Hello, my name is Rebecca and I work in the Business Development Group at Axxam. Today, I'm going to talk you through who we are and the services we can offer you for your early stage drug discovery projects. Axxam is an innovative partner research organisation, or IPRO, based in the Open Zone Science Park in Bresso on the outskirts of Milan in northern Italy. As an innovative partner research organisation, we differ from a CRO in our more personalised approach towards our clients. Our highly qualified and experienced scientists, well known for their passion for innovation and science, will channel their vast knowledge and experience into your research project and become an integrated part of your research team. We specialise in generation of tractable hits through high-throughput screening campaigns, relying on our strong experience in generating good assays and accessibility to novel compound libraries. We can develop from scratch functional in vitro cell-free and cell-based assays on demand, but we can also give you access to a long list of our ready-to-use assays. Our long experience in validating and optimizing assays to industrial standards means we have a solid background in assay transfer, either from or to your laboratory. We perform screening campaigns with several readouts such as fluorescence, luminescence, radiometric, high-content imaging, and electrophysiology, using either the assays developed at Axxam or assays you transfer to us. Screening can be conducted using our compound collections or your own compound libraries. We also take care of screening follow-up activities. All our services are offered in a plug-in, plug-out mode, so you can decide to benefit from our entire hit generation process or just take advantage of specific services. Our molecular biology unit at Axxam can develop assays from scratch, including designing the constructs and using cutting-edge technologies of genome editing to generate the required cell lines. We also offer messenger RNA expression analysis and TaqMan-based medium throughput screening to identify compounds that modulate transcript levels. We are experienced in developing functional cell-based assays for all target classes, such as G protein-coupled receptors, ion channels, nuclear hormone receptors, solute carriers, and many more. We generate assays in 384-well format for high-throughput screening purposes or as secondary and orthologous assays for compound profiling and hit-to-lead activities. We also have experience studying important biological processes such as autophagy, mitophagy, lysosomal storage disease, and phagocytosis. Our unit dedicated to the use of human-induced pluripotent stem-derived cells allows us to develop and study more physiologically relevant disease models, also in combination with optogenetic readouts. Here we routinely use human iPS-derived neurons and cardiomyocytes, as well as co-cultures. Our expertise in high-content imaging enables us to develop and perform phenotypic assays and follow pathways including the autophagy process, endocytosis, protein degradation, neurite outgrowth, and translocation, and many more. Our dedicated high-content lab can generate assays and profile compounds on a small scale or in a high-throughput manner. As previously mentioned, our expertise in developing cell-based assays means we have a large portfolio of ready-to-use assays developed in-house. You can access these for screening and profiling purposes, or you can purchase these for your own in-house activities. In addition to cell-based assays, we have a wealth of experience generating cell-free assays for several targets, including protein-protein interaction and enzymatic assays, such as oxidoreductases, transferases, hydrolases, lipases, isomerases, and ligases. Our biochemistry unit can perform the entire process from the production, extraction, and purification of the protein to characterizing protein activity and scale up for obtaining quantities of protein suitable for a high-throughput screening campaign. We carry out screening activities at both our headquarters in Breslau as well as at our site in Constance in Germany. Both sites are fully equipped with multiple automated screening stations with different detection systems. The screens can be conducted using all optical readout modalities in 384 and 1536 well format, including flash and glow luminescence, fluorescence, such as fluorescence intensity, fluorescent polarization, and TR-FRET, as well as others. We also perform high-throughput radiometric and high-content screening and medium-throughput TACMAN-based screening. To generate hits in a high-throughput screening campaign, you can choose to provide us with your own compound library or access the compound libraries available in Axxam. These innovative libraries have been assembled by incorporating compounds with structural and molecular properties suitable for hit discovery and hit-to-lead programs, avoiding unwanted reactive chemical groups, frequent hitters, toxicophores, aggregators, and other undesirables. Axxam's proprietary AXXdiv 4.0 library has around 325,000 small lead-like molecules with the aim of covering the most diverse space in terms of chemical scaffolds. The goal is to ensure that the resulting screening hits are readily tractable starting points for further optimization. This library is divided into 4 screening subsets with no overlap between them. Each subset can be accessed independently. Thanks to our strategic alliance with Cymeris in the Netherlands, the SymeGold compound library is also now available to you at Axxam for screening. This includes around 75,000 small lead-like molecules. We can also provide support for your hit follow-up activities by generating ad hoc assays such as orthogonal selectivity assays, and by further regular testing of compounds. This can be done as a one-shot activity or on a regular basis to support your hit-to-lead program. Our electrophysiology unit can validate compound activities for electrogenic targets. The lab is equipped to validate new cell lines, to perform compound profiling activities, as well as study the mode of action of the compounds or structure-activity relationships, with different levels of automation, either in a low or high throughput manner. In addition, we have a solid experience in organella electrophysiology, such as patch clamp on lysosomes and nuclei. In addition to these early drug discovery services, we have formed strong partnerships with leading service providers to be able to offer you integrated drug discovery projects under a streamlined project management. Our highly experienced team here at Axxam is available to work with you as your trusted partner for any of these early drug discovery services described during this presentation. Whether you are a large company looking to outsource projects, a startup or virtual company needing access to highly skilled scientists and well-equipped laboratories, we can help you. Our team of professionals will consistently ensure your project receives the highest level of attention and engagement, no matter the size of your project. Please feel free to get in touch with us directly if you wish to discuss your project with our team. To find out more about our capabilities, you can visit our website, and to receive regular updates, you can subscribe to our newsletter and also follow us on LinkedIn. Thank you for listening, and we look forward to working with you in the future.